Hi, we're going to continue talking about Mendelian genetics, but now we're thinking about multiple genes who interact with one another to cause different phenotypes. So after this class, you should be able to analyze the results of an F2 dihybrid cross to propose a genetic model. The genetic model is proposing the number of genes, the number of alleles, the relationship between those alleles, which one is dominant, which one is recessive, or some intermediate relationship between the alleles, and if the interaction between those genes is additive or if there's an epistatic interaction. So let's start with an example. Let's say that you cross a true breeding tan lentil with a true breeding gray lentil, and you see that all of their offspring are brown. I ask you to propose a genetic model, and as someone who just watched my last video, you think, wow, this looks like a case of incomplete dominance, right? So maybe let's call the true breeding tan lentil big A, the true breeding gray lentil little a, because we know that true breeding individuals are homozygous with respect to that locus. So they must make heterozygous offspring, and this is a relationship of incomplete dominance of the A locus. That might work for now, but really what we have to do is see what happens in the F2 and see if this hypothesis, our proposed genetic model, still fits the results in the F2. So what would we expect in the F2 in this situation? Let's draw it out with a Punnett square. So here in the F2, we would cross with another one of these heterozygous brown lentils, and we would expect to see what we've seen a couple of times in this class, which is a 1 to 2 to 1 ratio of genotypes in the offspring, which would give us the tan phenotype here with the brown phenotype here and the gray phenotype here in a 1 to 2 to 1 ratio. So let's perform that cross and see what we get. See if it fits our model of a single gene that is incompletely dominant between these two alleles. And let's say that the actual results showed us this, that we observed a 9 to 3 to 1 ratio of 4 phenotypes among the outcome of this cross. Now, what kind of model can we use to explain these results? Well, we saw a 9 to 3 to 1 ratio in an F2 dihybrid cross last week, right? When we were talking about the yellow gene with the big Y, true breeding parental generation, and the green gene with the little y, and also the round and wrinkled phenotypes. And we saw that 9 to 3 to 1 relationship. But here it's a little bit different because this was two distinct phenotypes, the color and the shape, but here it's just the color. So how can we make a two-gene model that would perfectly fit the expected ratios, but where there's only one phenotype that's being modified? Well, we could first start to try to map the genotypes that we observed onto that 4 by 4 Punnett square of the results of the dihybrid cross. And you can do that by making a few educated guesses. So the first one that I always like to start with is, what do I do with this 1 here? Well, there's usually this one kind of special square, which is the case where for both genes, this individual is homozygous recessive. So let's call green the one that we saw the one of, the homozygous recessive. And now for the 9, I saw 9 brown lentils as a result of the F2. So here we can map those onto these squares. Now what these squares have in common is they have at least one big A allele and at least one big B allele. So here we're seeing a situation where maybe if you have at least one big A and big B allele, you have the brown color. And now exactly where I put tan and where I put green could be flip-flopped, but you can see what's happening here is that for the tan, the way I have it, the tan individuals have at least one copy of the big A allele, but no copy of the big B allele. And the gray individuals have at least one copy of the big B allele, but no copies of the big A allele. Now, We can think, what kind of molecular mechanism would explain this genetic ratio? And you can kind of flip back between this slide and the Punnett square on the slide before to see where I'm going with this. So what we're seeing here is a case of an additive interaction between two gene products. So what we know about the lentil is that there's chlorophyll inside, and there's sort of a layer around it that is transparent. And the lentil has two genes involved in producing pigment. There's one gene, which I have elected to call A, which makes the tan pigment, and there's one gene that I have elected to call B, which makes the gray pigment. Now, in this case, we are seeing that big A is completely dominant to little a. That is, if you have at least one functioning copy of the big A allele, you can produce this tan pigment. Also, big B is completely dominant to little b. If you have at least one copy of the big B allele, you can create the gray pigment. And when you have both, at least one copy of the big A allele and at least one copy of the big B allele, you make both types of pigment. Those mix together to result in this brown color that's deposited on the lentil. Now, what happens if you don't have a single copy of the big A allele? Well, then you can't make the tan pigment. But if you can still make the gray pigment, then you appear gray. Same, but opposite over here. If you don't have a single big B allele that is necessary for making the gray pigment, you don't make gray. But if you have at least one copy of the big A allele, you can make tan, therefore you're tan. And now, if you have neither, you don't have a single copy of the big A allele or a single copy of the big B allele, you can make neither type of pigment. So the green color of the chlorophyll is what shows through. You're not making any pigment in this lentil. So this is the molecular mechanism we can use to explain the additive relationship we observed that resulted in 4 phenotypes and a 9:3 to 3:1 ratio in this cross. But there are other interactions we can see between genes too, such as what if one gene was able to mask the phenotype of another one? And what would happen is that we would see fewer than 4 phenotypes produced in that F2 cross. And this is something that we call epistasis. The etymology of the word epistasis, epi meaning kind of on top, like epidermis, and stasis meaning to stop or hold. So you can think of one gene as on top of stopping the action of another gene. And here's my favorite example of this because it has cute pictures of dogs. This is a case of recessive epistasis. So here you observe from a dihybrid cross, you can observe black Labradors, yellow Labradors, and chocolate Labradors in a 9:4:3 ratio. And now we have to look at the Punnett square and get a little creative with how we're going to find the 9 and the 4 and the 3 in this Punnett square. So the 9 we've already seen. This is a situation where you have at least one copy of each allele. Here I've called the genes B and E. That one of the 3's is also the same. This is when you have at least one copy of, in this case, I made it the E allele, but you don't have a single copy of the big B allele. And now here is where we get into an epistatic relationship where there are 4 individuals that I've drawn here that are little e, but essentially the genotype at the B locus doesn't matter. But the thing that these all have in common is that they are little e, and that's an epistatic relationship. So what molecular mechanism could explain this genetic ratio? Well, now instead of thinking about 2 gene products that contribute to one color, like we saw before, let's think of it as a pathway. So we can think that there's some colorless precursor molecule, and then there's enzyme E that I've designated by the E gene that makes this dark pigment called eumelanin. And now there's another gene product that encodes an enzyme that's going to densely deposit that black pigment. So if this dog has at least one copy of the big E allele, it can make the eumelanin. And if it has at least one copy of the big B allele, it can densely deposit that eumelanin, so the dog appears black. If the dog has at least one copy of the big E allele, it can make the eumelanin, But if it doesn't have at least one copy of the U melanin allele, it can't densely deposit it, but it still can somewhat deposit it, and so it looks like this brown color because there's some eumelanin being produced and being deposited, just not as densely as it is when there's this B allele around, the big B allele. And in the last situation, when there's not a single big E allele for the enzyme that can make the eumelanin, well then the dog is not making the dark pigment at all. Therefore, it doesn't matter what the genotype is at the enzyme that would deposit the eumelanin, or in other words, little e is epistatic to the B gene. The little e allele is masking whatever genotype the dog has here, and that dog will appear yellow. We call this recessive epistasis. There is a recessive genotype that is acting epistatically to the genotype at a different locus. We can also observe dominant epistasis. In here, you would observe a ratio of 12 to 3 to 1 of 3 different phenotypes among the offspring of the dihybrid cross. And now here, you can see where I have mapped those 12 to 3 to 1. The 1, again, I'm saying it's kind of the special case of the homozygous recessive. I have chosen this as the 3, where again, it's the little e allele, but at least one copy of the big B allele. And for the 12, I've chosen anything that has at least one copy of the big E allele. So what molecular mechanism could describe this pattern in the F2? Here we can imagine another pathway type situation of an enzyme that creates a pigment and an enzyme that deposits a pigment. But now the situation with the depositing pigment is flipped, such that it's the recessive allele that's responsible for depositing the pigment, and it's the dominant allele that does not deposit the pigment. In other words, the dominant allele is the one that's acting epistatically, that is epistatic to the other gene. So let's say that you have some green starter pigment, and there's an enzyme that turns the green pigment to yellow. So if you have at least one copy of the big B allele, that enzyme, then you can produce the yellow pigment. If you do not have at least one copy of the big B allele, you do not produce the yellow pigment. Now, so for this top example, you have the green pigment that can be turned into the yellow pigment because there's at least one copy of the big B allele. But because the dominant big E allele prevents the depositing of that pigment, then this squash was the example, would still appear white. So the genotype here masks the genotype here. This dominant allele stops that pigment from being blocked. And you see the same thing here. The presence of that dominant allele masks the genotype here. It doesn't matter. The fact that there is one big E allele here means that that pigment will not be deposited and this squash appears white. Now, if the genotype is homozygous recessive for the allele that does deposit the pigment, now the genotype at this other locus does matter. So if this individual has at least one copy of the big B allele, they can produce the yellow pigment and it can be deposited so they appear yellow. This individual does not have at least one copy of the big B allele. It does not produce the yellow pigment, and that yellow pigment is not deposited, or sorry, the yellow pigment is not made, but the green pigment is still around and can be deposited. So this step gets kind of skipped over and the green is deposited, resulting in that 12 to 3 to 1 ratio among the offspring. Another epistatic pattern you can observe in the results of an F2 cross is a reciprocal recessive gene. And this would show up as a 9 to 7 relationship, which is seen in these flowers here, which would have 9 of the offspring in any case where there is at least one big B and a big E would appear pink. And if there is not both a big B and a big E, then the flowers would appear white. The molecular mechanism that could explain this genetic ratio is again a pathway type analysis. But here it's reciprocal and recessive. So in this case, for any flower that has at least one copy of the big B allele, which I've designated as an enzyme that turns a precursor into a second precursor, and at least one copy of the big E allele, which I've designated as an enzyme that turns the precursor into the pigment, then those are the flowers that will appear pigmented. On the other hand, if any one of these enzymes are lacking, then those flowers will all appear white because either the first precursor can't be made, so it doesn't matter what the genotype is at the second enzyme because it requires that precursor, or the first precursor is made, but the second enzyme isn't functional, so it can't produce the purple pigment. Or both could be non-functional. Therefore, the precursor and the pigment is not made. This is reciprocal epistasis because in this case, we say that the little b is epistatic to the genotype here. This is the genotype that's masking the genotype of the big E, or big E, little e. But in this situation, it's the little e genotype that's masking the genotype at this locus. So it's reciprocal and it's the recessive genotype that's doing the masking. And finally, you can have a situation where genes act redundantly. So here's an example of the shepherd's purse seed pouch that sometimes takes on this kind of triangular shape and sometimes takes on this more oval shape. So in an F2 cross, which you could see here, is a ratio of 15 to 1. So again, our 1 is the special one that is the homozygous recessive for both genes. That is the only one that is going to make this oval shape. The molecular mechanism that could explain this genetic ratio is more straightforward. Essentially, these two pathways act in parallel and they're redundant to one another. One of those pathways could be totally broken, and the plant could use the other pathway to still get the same phenotype. So in this case, in the first case, we have both pathways working normally. You get the triangle shape. In the second case, you could have the little b genotype, but that would be fine because the cell would just use the E pathway to create the triangle shape. In this case, you could have the little e genotype. That would be fine. The individual would just use the B pathway to make this shape. It's only when both of these pathways are broken, when there are non-functional alleles in both, that you get the ovule shape. So hopefully, going through those mechanisms and those ratios have prepared you to analyze the results of crosses yourself and start to propose genetic mechanisms.